Welcome to Government of Andhra Pradesh Commissionerate of Collegiate Education. I am Mrs. Syed Wajiha Tahsin, lecturer in biochemistry, SRR and CVR Government Degree College, Vijayawada. Today we are going to discuss about oxidative phosphorylation, that is synthesis of ATP. Dear students, in the previous class you have learned about components of electron transport chain and how these components are arranged in the form of four complexes, that is complex 1, complex 2, 3 and 4, which are involved in oxidation of NADH plus H plus and FADH2 and the movement of electrons towards oxygen for the synthesis of ATP, for the synthesis of water, and simultaneously protons are pumped towards the intermembrane space. Now you are going to learn how this electron transport chain is coupled to the synthesis of ATP by the process called oxidative phosphorylation. Before going to learn about how the mechanism of oxidative phosphorylation takes place, first we will learn about the structure of F0 F1 ATP synthase. So this is F0 F1 ATPase is an enzyme system that is present with the complexes in the inner mitochondrial membrane or thylakoid membrane in the chloroplast, which is involved in the phosphorylation of ADP for the synthesis of ATP. So this ATP synthase is an enzyme that catalyzes the synthesis of ATP. As you know that ATP is the energy currency of the cell and it is the high energy compound, high energy phosphate, so which is synthesized usually. Previously what you have observed? You have observed that in glycolysis or TCA cycle, so the substrate itself is phosphorylating ADP to synthesize ATP, that is called substrate level phosphorylation. Now the synthesis of ADP is carried out just by donating the phosphate from the substrate and phosphorylating ADP to ATP. Now you are going to see how this electron transport chain is coupled in the synthesis of ATP with the help of F0 F1 ATPase enzyme system. So the overall reaction carried out by ATP synthase is ADP plus Pi plus 3 protons gives rise to ATP plus water. So this complex, that is F0 F1 complex, is located in the inner mitochondrial membrane and thylakoid membrane. So now you will see the structure of ATP synthase. The structure of ATP synthase, you have two components basically, that is F1 and F0. F1 is a knob-like structure and F0 is the stack structure. This F1 knob-like structure is protruded from the inner membrane of mitochondria towards the matrix, and F0 is the stack-like structure that is located in the inner membrane itself. So this F0, which is the stack-like structure, which is rotated by F0, which is rotated with the help of the stack structure, that is F0. So F0 causes the rotation of F1 and F0 F1 creates a pathway for the movement of protons across the membrane. In the structure of F0 F1 ATPase, you can observe the F0, that is stack-like structure, and F1, that is knob-like structure. So this usually this structure, which is located in the inner mitochondrial membrane, is of 100 nanometers in diameter. And coming to in the F1 stack-like structure, you have alpha 3 beta 3 subunits which are having the catalytic activity of ATPase. So with that you have delta and gamma subunits. Completely 3 alpha subunits, you can see in the diagram, 3 alpha subunits, 3 beta subunits, gamma and delta subunits are existing in the F1. Coming to F0 structure, you have the membrane located, that is which is inserted in the membrane. You have the subunits called A, B, C. So many C subunits, that is nearly from 6 to 15 C subunits exist depending on the type of the organism. You have many C subunits to which A and B subunits are also attached. So this complete F0 and F1 structures are involved in the transportation of H plus ions, and movement of H plus ions causes the movement of the gamma like structure, which causes the movement of these alpha and beta catalytic subunits, thereby causing the synthesis of ATP. The F1 knob-like structure contains the catalytic subunits, that is ATP synthesizing subunits, 3 alpha subunits, 3 beta subunits, and it also have gamma and delta subunits. So this F1 is actually it is a water soluble part, and which is protruded from the membrane into the matrix, and it can catalyze the synthesis of ATP. The subunits alpha and beta makes the hexamer with the 6 binding sites. The 3 of them are catalytically inactive, only they can help in the binding of ADP, and other 3 subunits catalyzes the synthesis of ADP. The other F1 subunits are gamma, delta and epsilon, which are part of the rotational motor mechanism. They are involved in the rotation of these beta and alpha subunits. So gamma subunit allows the beta to go through many conformational changes. So there exist 3 different types of conformational changes, that is closed, open half and open states, that allows the binding of ADP and release of ATP. So the F1 particles usually you can see, which is having a diameter as we have earlier discussed, and it can be seen in the TEM, that is the transmission electron microscope, by the negative staining process. Coming to F0, that is the stack region. F0 has a proton channel which spans the membrane. So it forms a proton pore. Here you can see the proton pore. F0 is hydrophobic in nature, and which is actually inserted in the inner mitochondrial membrane. So it has a ring of multimeric helix protein, which is C, which is made up of C subunits. The helix protein undergo a conformational change. Usually when proton is pumped here from the inner membrane space, that is intermembrane space, to the matrix, when protons come back from the intermembrane space into the matrix, what happens? These subunits are got rotated, causing the spinning of the F0 subunit, which then also affects the conformation of F1. So these C subunits will spin by the movement of protons, which also allows the movement of the conformational change of F1, resulting in switching of states of alpha and beta subunits. The F0 region of ATP synthase is a proton pore that is embedded in the mitochondrial matrix, as we have discussed. See, movement of protons from intermembrane space into the matrix, causing the rotation of the C subunits, which causes the rotation of alpha and beta subunits, and their change in conformation, which allowed the synthesis of ATP by binding of ADP plus PI. So this F0 stack region, as we have earlier discussed, consists of ABC subunits. C are around from 6 to 15 subunits exist. Usually 6 C subunits make up the rotor ring, and the number can be changed from up to 15, and the subunit B makes the stack connecting the F1. This part of the enzyme is located, as we have discussed, in the inner mitochondrial membrane and couples the proton translocation to the rotation that causes the synthesis of ATP in the F1 region. So F0 this region is very sensitive to oligomycin, which is an antibiotic that binds in the channel and blocks the H plus passage, thereby inhibiting the ATP synthesis. So here movement of protons is very important for the synthesis of ATP. So how the movement of protons causes the synthesis of ATP is the theory of ATP synthesis was given by Peter Mitchell, that we'll discuss by this chemiosmotic theory. So the chemiosmotic theory, which proposes the synthesis of ATP by the proton motive force. This theory was proposed by Peter Mitchell in 1960s, and he got Nobel Prize in 1978 with the other British biochemists John Walker and Paul Boyer from the US. They shared the Nobel Prize for Chemistry in 1997 for their work on this reaction of ATP synthesis. So what is the complete chemiosmotic theory, that we will see in the coming slides. So actually in the inner mitochondrial membrane you have complex 1, complex 2, complex 3, complex 4, which are located like in this order, and finally electrons are transported from NADH or FADH2 to complex 3, and from complex 3 the electrons are moved to complex 4. Finally they reaches to oxygen for the synthesis of ATP. Simultaneously, when the electron moves, the protons are moved into the intermembrane space. The protons that are accumulated in the intermembrane space causes the development of acidic charge towards the intermembrane space, and acidic charge, and not only that, so there is a proton motive force generated. So this due to this proton motive force, actually protons flow back again from the, as we have discussed, the F0 F1 ATPase structure. Protons move back through this structure into the matrix, simultaneously causing the phosphorylation of ADP to ATP. So here the proton motive force is very important force, which helps in the formation of ATP from ADP. So this is Peter Mitchell theory. The proton concentration gradient serves as the energy reservoir for driving ATP formation. So electron transport through the complex of respiratory chain causes uptake of protons from the matrix and release of protons into the intermembrane space, so which generates a proton gradient. So the movement of protons makes the intermembrane space positive and slightly acidic relative to the matrix of mitochondrion. So as you know that from the matrix protons are pumped to the intermembrane space. So here there is a relative difference in the proton concentration, which allowed for the proton motive force. The difference between the two sides of the membrane is called proton motive force, and it can drive proton back from the intermembrane space into the matrix through the F0 F1 synthase enzyme. So this Mitchell theory explains that the proton motive force is the energy of proton concentration gradient. Protons that are translocated into the intermembrane space by electron transport flow back into the matrix via ATP synthase. So as protons move back into the matrix through ATP synthase, the energy stored in the electrochemical proton gradient is used to make ATP. So the transmembrane protein ATP synthase catalyzes the phosphorylation of ADP with phosphorylation of ADP in a reaction driven by the movement of protons across the inner membrane into the matrix. So the proton flow through the enzyme causes a rotation of protein subunits in the F0 F1 ATP synthase, which leads to ATP synthesis from ADP and phosphate. So this proton movement causes the phosphorylation of ADP plus Pi with the help of the catalytic beta subunits. So here you can see we have taken only the beta subunits and this is the gamma subunit which helps in the rotation of these subunits. So when protons are pumped, so usually the beta subunits exist in three different conformations: open, tight and loose. So in the open configuration, they bind to ADP plus Pi, or in the loose or open configuration they bind to ADP plus Pi, and in the next when it is rotated, usually it rotates 120 degrees and 120 degrees. So complete rotation 360 degrees. Every 120 degree rotation in the gamma subunit causes the rotation of this beta subunit and causes the conformational change. So when this open structure is converted into tight structure, ADP plus Pi is converted into ATP, and so when the tight structure again it is converted into open, which causes the release of ATP. So here you can see TO open structure. So three different types of structures by 120 degrees rotation is possible. So every 120 degree rotation, the open loose structure is converted into open structure is converted into tight structure. In this way, the three conformational changes are possible. So because protons when moved from the F0 F1 ATPase, especially from the stack-like structure, when protons are pumped, which causes the rotation of the C subunits. The C subunits when gets rotated, they causes the rotation of gamma subunits. When the gamma subunits got rotated, they usually in the beta subunit you can observe the conformational changes. So through these conformational changes, your ADP is converted into ATP. In that way, protons